whatever the was
Mm-hmm.
of the ethylene and the water.
Okay.
What I was gonna ask you is, so on Macmo, do you want us to like kind of make like a rough estimate of a transition state?
Yeah.
Okay,
Yeah, you just give it your best go. And I kind of showed you vaguely what it should look like, right?
yeah, so should I like be putting the water molecule like kind of like basically right there?
Yeah.
Okay, well, because I did that and I was just wondering if that was like the right thing to do, and I just
Yeah. And then you got to do ethanol too, so you get products.
yeah.
You want to come join? There's room.
You say you always want to do okay. Who else? You get that. Library. Okay. Maybe just wake up too late.
David will be here soon-ish, I'm sure. So we just took a midterm. Obviously, I haven't gotten it back to you yet. But the next, so for the rest of this class, so the labs, there's a couple more like wet labs we can do for more practice, but it's gonna be a lot of the same stuff we've already done. You know,
Kind of just
set up your glassware, reflux, distill, do a recrystallization, do a melting point. So I think, so given this is week 7, I think we can take the time in lab today to finish up the computational stuff. And I can hang out, we can go over to the library, and I'll just hang out over in the library.
Yeah.
Answer questions as they come up. And then I'm kind of, we have one more good paper lab on 2D NMR, which basically, if you remember like the DEPT proton NMR, where it was like testing two things coupled together. 2D NMR works like that, except you actually get a 2D graph out of it.
Okay.
And so your signals are actually in the third dimension. That signals show up. We usually just use a heat map to show, like, you know, red is a really intense signal. It looks more like a topographical map.
So that'd be 1H or 13 carbon? Or is it both?
Both. You can do both. You can
okay.
do it. And so then you can say this proton signal is coupled to this carbon signal. It allows you to say with a lot more certainty what signals go where. I actually just had those pulled up just so you can see what they kind of look like.
So it kind of gives you like a better guess to like what you're actually looking at on the signals.
Yeah, so you get stuff that looks like this. So if we look at a 1D NMR, proton NMR, it looks like that. If we have two frequency dimensions, we can see what proton signals are coupled with other ones. So anything that's on the direct axis, which is going to be this direction. See how everything is kind of symmetrical around the line right here?
Right, yeah.
The ones that are not on that line, these are mostly just the protons coupling with themselves.
Yeah.
But anything that shows up not on that diagonal is a proton coupling with the proton next door. So it allows you to say that this signal is coupled with this signal. So those two protons must be adjacent to each other. So basically remove some of the guesswork from, well, I think that this CH2 is next to that CH2, but I'm not sure. This allows us to, exactly. So I think we'll do that one next week. And then I think we'll start, there's one more, I guess the other one that is kind of a fun one to have you guys troubleshoot is extracting caffeine from tea leaves. So it turns out there's not that much caffeine in tea leaves. So it's actually kind of a tricky process to get decent yields and get a measurable amount of caffeine. So that's kind of that'll, I think, will be our last wet chemistry one. And then we'll do some sort of find a cool OChem paper and present it for the lab final. And so that last couple of weeks will be that, like, pick your paper with me on week 11 and then present it week 12.
I went to Davis to shadow my sister for some of her classes.
Yeah.
And it was a biomaterials class.
Really,
really cool.
Nice.
And I was honestly thinking that I might maybe pull something from that with a little bit of.
Biomaterials and bioengineering, there's a lot of really cool stuff. Was she doing like tissue engineering, or was the class talking?
All bio.
Okay.
Materials. Well, she's doing a biomedical engineering, so.
Okay, yeah, so the bioengineering section of so bioengineering and chem engineering were the same department at CU. 15 years ago, bioengineering was enough in its infancy that they really were part of the chemical engineering departments. Now it's grown a lot. But that's really fascinating. Like there's really cool stuff they can do, like use OChem to build a 3D lattice of a material and then grow the cells in the material. And if you start from stem cells with the right chemical messengers and with the right physical framework, you can produce things like cartilage that then you could then use to make like a new kneecap or things like that.
We were actually looking at that. In
Yeah.
Manipulation of extracellular matrix. So basically just that.
Yeah, exactly. And so were they using, do you remember if they were using PEG as their, which is polyethylene glycol, was what was used to make like the 3D matrix for the cells to grow in?
So they were yes, they did. And they also kind of just took that kind of more, I guess you could say, natural approach to it by like actually introducing certain proteins.
Oh yeah.
Just like
okay.
Modified like
cool. Yeah.
All that kind of stuff.
That's really cool. That's going to be a really big field, especially as we have more and more need for rebuilding knees and enamel on teeth and things like that. Biomaterials is pretty cool.
So that kind of inspired me. I was like, oh wow, that's like really cool.
Yeah. That'd be awesome. That's a really cool field. So then as far as the material we've got left to cover, we've got carboxylic acids and derivatives, and then alpha carbon chemistry. And then whatever else we have time for, we can kind of pick a couple of topics towards the end, depending on how lectures go and what everybody's into. There's a decent chapter on polymers. And I don't really like the approach of organic chemists getting into biochemistry because I don't feel like they approach it from the right point of view, because they don't do anything like biological with it. So it's more about like, here's a reducing sugar and this is a non-reducing sugar, and those will be important later. It's not really actually getting much into the actual applications or biological systems. It's biological molecules, not biological systems. So it's a little bit weird. So, but we'll figure that out as we get closer. So anytime we added a new functional group, we always have this slide, right? Here's a bunch of random molecules that have this functional group. Although some of the carboxylic acids, the simple carboxylic acids are actually kind of interesting. So acetic acid and vinegar, the smell of butter gone off is actually butanoic acid.
I've never smelled that.
You don't smell it on the butter itself, but like really old butter, if you try to cook with it, then you get it. It's got a pretty low vapor pressure until you heat it up. But like dirty socks, anything that you would like describe as like pungent, a lot of times is in carboxylic acid, lactic acid, citric acid. We use like that's they use citric acid as like the coating on sour candies. Right. But then obviously it shows up in lots of other stuff. Retinol is the brand name, not retinol. What's the brand? There's a brand name for Accutane.
So yeah, I recognize that.
I'm pretty sure that that's Accutane. Aspirin. There's our acid group. There's our ester group. Aminosalicylic acid apparently is used in treatment of tuberculosis. I didn't know that until I made this slide. So, a lot of biological molecules though, because we do wind up, anytime we've got these oxygen-based functional groups, all of these biological molecules that we use as energy most of them start as an ester or an alcohol in some form, and then as you oxidize it down, that's how you generate the energy. And so we see a lot of these as sort of either byproducts of fermentations of microbes, or is it sort of intermediates in our body, lactic acid in particular. But a lot of these, like hexanoic acid showing up as the odor of dirty socks, it's actually the bacteria on your feet producing hexanoic acid because of the conditions they're in that make that molecule. So we see a lot of them show up. These are wind up being really important molecules in biochemistry. Naming these, we've danced around it before, but officially, you drop the E, name it like it's an alkane, drop the E and put oic acid. So very similar to aldehydes, where you drop the E and add Al. In this case, you drop the E and add oic acid.
And that's going to take the highest priority of
exactly.
Before.
Yeah. There, if you do wind up naming this as a prefix, a lot of times you'll see carboxy. Or a carboxyl group is the prefix way of describing that. And then if you deprotonate it, you call it carboxylate. Any time you have a deprotonate form, it's actually when you first learn naming acids in Gen Chem, like, oh, you learned your polyatomic acids first, and then like, okay, if it ends in 8, you drop the 8 and we put ic acid. It's actually that's kind of backwards. It's more that we name the acid first, and the deprotonated form turns into an 8. So basically, instead of butanoic acid, it is deprotonated, it's butanoate. You do keep the O in there, so it winds up with sometimes you might have these names being a little bit wonky. But that's how you get benzoate, butanoate, acetate, obviously is going to be deprotonated form here. Formic acid. So these are the most common names. The ones you really need to worry about, like pretty much nobody uses butyric acid or propionic acid anymore. It's propanoic acid and butanoic acid, butanoic acid, I mean. But benzoic acid, acetic acid, and formic acids that we still use those common names. And the deprotonated form of the formic acid would just be formate. But benzoate shows up all over the place because it's not toxic, but it's a decent preservative. So pretty much anything packaged has sodium benzoate added to it. It's not like, health food necessarily, but it's not bad for you. And it's a pretty good way to make sure that bacteria and microbes don't ruin your food. If it's a dioic or if it's a diacid, we just say dioic acid, pentane dioic acid. The trick with dioic acids is that the chain always has to start and end, right? With an acid, and so then that can make your dominant carbon group a little bit weird. But for the most part, the dioic acids that are actually important pretty much all have common names. And again, they show up a lot in biochem. Did you learn the citric acid cycle and electron transport chain? Like a lot of these, like oxalate shows up in there, succinate. If you add a nitrogen on in the right spot here, you get glutamic acid, the amino acid. Yeah, we see most of these in their deprotonated form, though, are the most common form in biochem, because under biological conditions, most carboxylic acids are going to be deprotonated. So generally speaking, we don't name them as the acid. We don't call it oxalic acid. We call it oxalate. Yeah. Oxalic acid, though, is actually the main active ingredient in barkeeper strength. If you ever use that to clean stuff. For dishwashing and stuff like that, oxalic acid is why it works so well. It's a little bit of an abrasive, but it's also a slight reducing agent. So, like I said, a lot of common names with these acids, especially with the dioic acids.
Want us to know the common names?
I'm not gonna test you on these. That's something we'll save for a biochem class,
Okay.
when you have to memorize them
Yeah.
and where they go into a pathway. For now, no, don't worry about it. Other than, the common names. Acetic acid and benzoic acid.
I mean, yeah.
Yeah, like we are used to those ones. And then, if you were gonna pick one of these, oxalic acid. But you could technically name oxalic acid as ethane dioic acid. Would not be wrong. Yes, yeah, most people know it as oxalic acid though. Let's see. Let's just do the two big ones on the left, or on the right, I mean. It's gonna be hexanoic acid, and then it's gonna be 3,4 tetramethyl. Then here, the only trick is our longest carbon chain that starts with the acid. So it's only gonna be pentanoic acid. And then with the propyl group, so two propyl. Yep. Easy enough, just more of the same, we just added acid to our toolbox. In terms of properties of carboxylic acids, they tend to have really high boiling points and melting points, mostly because carboxylic acid group on its own has really strong intermolecular forces. Just within the carboxylic acid group, it's got 3 polar bonds, including 1 hydrogen bond. So it winds up forming really strong intermolecular forces. If you look at ethanol, or even something that's with more heavy atoms like isopropanol, you look at their boiling points. Acetic acid's got a much higher boiling point. David?
Good morning, sorry.
No problem. Right, so when we're naming, or when we're looking at these properties, we generally, they all tend to have the same general trend, right? Within the same functional groups, you're looking at what's the highest molecular weight. But then if you add more polar functional groups, that's obviously going to get higher melting point, higher boiling point. I jump back to, aspirin, acetylsalicylic acid. The melting point for that one, it's still only got 1 carboxylic acid. It's got an ester, which is somewhat polar, but not super polar. And then the overall size of this, that's why it doesn't even melt. I want to say melting point is like 120 Celsius. Compared to like benzaldehyde, which is almost as large as a molecule, or vanillin for that matter. Vanillin is almost as large as a molecule, but it doesn't have the acid group. And so vanillin melts, actually, I don't remember if it's solid or room temperature, it's close to room temperatures where it melts. So just like we'd expect, the reason that they're acidic is mostly because of resonance. And so if we look at, and if you think back to when we first started talking about acidity of molecules, we said, look at how stable the deprotonated form is. Because the deprotonated form is going to be the one with the charge. And so inherently we would think the deprotonated form is less stable. And we're right for the most part, but it's stable enough that these are considered an acid because of resonance. The deprotonated form, you can resonate put the charge the other direction. So that's still just as unstable, that the fact that we've got two equal resonance structures makes it a lot more stable than it would otherwise be. A lot more stable than, say, deprotonating your average alcohol. Beyond that, so pKa is typically between 4 and 5. So more acidic than water. Because pKa of our pH of water is 7. So at pH of 4 or 5, or sorry, at a pH of 7, they're going to be predominantly deprotonated. If you remember how we first started talking about acids and bases back in probably September even. If we look at the buffer equation, the difference between your pH and your pKa raised to the 10th power gives you your ratio of deprotonated to protonated. Right, so if I rearrange this 10 to the pH minus pKa, so the difference between your pH and your pKa, 10 raised to that difference is your ratio. And then so if you're at neutral conditions, if you're at pH of 7, and we're talking about benzoic acid that's got a pKa of 4.19, that's a difference of almost 3 pH units. Yeah. So your ratio of deprotonated to protonated is about 1,000 to 1. Or in that ballpark anyway, within a factor of 10. Which is why when we were talking, like I said before, when we were talking about biological applications of these, or any of these acids that show up in biological systems, for the most part, are close between about 7.2 to 7.8 pH. So they're almost always going to be present as their deprotonated form in a biological system. Exception, of course, being other life forms that don't have I think actually all life on Earth is pretty close to neutral. Even if it can live in acidic conditions, the interior of the cell matrix is close to neutral. The exception being in digestive systems, some animals like humans, mammals tend to have pretty acidic digestive systems. So I want to say the pH of the stomach is about 2. So most of these would be protonated in the case of being in the stomach, which winds up, if we get a chance to talk about protein structure at all in this class, it winds up making a difference for how the proteins fold. When your glutamic acids are all protonated versus deprotonated, for instance, your proteins are going to fold differently at different pHs, predominantly because you're going to change all those intermolecular forces by protonating and deprotonating different molecules.
Deprotonated, it's going to change hydrogen bonds between different amino acids.
Yeah, so just as a real general case, like if you look at an amino acid that's got a a basic side chain, like, is it proline, I can't remember all the amino acids off the top of my head.
I can't either.
Proline, I think proline and histidine are the basic ones. I don't remember their structures, but basically they both have a nitrogen as part of the side chain. And under physiological conditions, normally that's going to be protonated. And so you're going to wind up with a positive charge here. And if you're relying on, and then you have this big long chain of amino acids. And then on this side, if you have a glutamic acid that's deprotonated, it's going to make these really strong attractive forces between these hydrogens and the glutamic acid. If you protonate this one by putting it into acidic conditions, all of a sudden it falls apart. So actually there are enzymes that are actually specifically evolved to work in our stomachs, because they need to have a different structure because of the acidity levels. If you take, there's proteases in our stomach, proteases that break down amino acid bonds, peptide bonds. Those proteases only work in the stomach. Your cells actually have similar proteases inside that are actually evolved to work at pH of 7. To keep basically so that proteins that get into your cells have a finite lifespan. They get periodically broken down and recycled for parts, basically. But that's very different conditions than your stomach. And so we actually have different versions of the same enzyme, basically, one for the stomach and one for the rest of the body, based on those pKa's.
Yeah, because I think like the big thing that all life has in common, right, is like DNA and RNA replication, and I think the ideal ranges for like those enzymes tend to be close to neutral.
Yeah, we share pretty much all of our DNA and RNA polymerases all share the same ancestry.
Yeah.
And they tend to be right around 7.
And so you have to like maintain that, even if you're living next to a hydrothermal event.
Exactly. So the interior of the cells still needs to be at that same pH, even if the exterior is wildly different. So when we start looking at other things beyond, in general, the more resonance you're going to have with your deprotonated form, the more acidic it's going to be. The easier it's going to be to pull that hydrogen off. So they're all in the same ballpark. The main thing is getting that first resonance structure. But in general, as you add more resonance, the more acidic it's going to get. As you pull electron density away, the more electron withdrawing groups you have, the more acidic it's going to get too. And that's because you can kind of think of it as basically the more electron density you have on this oxygen, the tighter the hydrogen is going to be stuck on that. By pulling electron density away, either with resonance or by adding electron withdrawing atoms, you're going to make it more acidic by doing that. We can see that too with if we look at the acetic acid and we add progressively more chlorines to it. Chlorine's not even that electronegative compared to fluorine, but you wind up with it dropping 2 full pH units just by making it chloroacetic acid. And then if you make it trichloroacetic acid, now we're almost into strong acid territory. Anything with a pKa less than 0 is going to be considered a strong acid, like hydrochloric acid. So this is only, 10% off of being all the way down to the similar level of acidity as hydrochloric acid.
I never knew that was the official cutoff for strong. I thought I had no idea.
And so yeah, it's because so if you think about it, anything that's got a pKa less than 0 means it's a better acid than water is.
Yeah.
And so you can protonate waters, you can donate the protons to the water 100% of the time effectively once you get past the pKa of 0. And bases, it's a little bit different because bases you have to deal with ionic compounds that are just straight up dissociating rather than thinking about it in terms of proton donors. Strong bases tend to just be anything that's a hydroxide with a high solubility. Oh, there you go. I forgot to put that on the next slide.
Why do you even need slides?
Right, right, all up here. So let's do a practice with that, just like I did there. If you've got a pH, if this solution is buffered to a pH of 5.76, what's the ratio of acetate to acetic acid? And again, pKa for acetic acid was 4.79. No, 4.76. That makes that chosen pH make a whole lot more sense.
That's so nice.
Yeah.
So 5.76 equals 4.76 plus log of that ratio. So again, most, especially on the biochemical side of things, and even in the even to some extent in biochem, they use this equation a lot. They just call it the buffer equation because they use it to make a buffer. If you want to make a solution and keep it at a certain pH, this is what you do to figure out, like, okay, I need to add this many moles of sodium acetate and this many moles of acetic acid, and then I can get that right ratio. But the other way of thinking about it is, it tells you at a given pH what is the ratio, not just what do you need to do to make the buffer, but what form can you expect to find the acid in? Right, so in this case, we're going to get 10 equals A minus over HA. Right, and if you're ever unsure about if you missed a negative sign in there somewhere, it's supposed to be, this is obviously give you 10 to 1, the way it's written, deprotonated to protonated, so you have 10 times more on the deprotonated form. If you're ever wondering if you missed its negative sign in there somewhere, think about, is your pH more basic or acidic? If you're more basic, the deprotonated form should be more common because there's fewer H pluses around. If you're more acidic than your pKa, you should have more of the HA, more of the protonated form. So it's just as far as doing like a reasonableness check when you actually think about this, just think about it, okay, if I'm at my pKa, it's 50-50. It's a 1-to-1 ratio. If I'm more acidic, I'm going to favor the protonated form. If I'm more basic than that, I'm going to favor the deprotonated form. Because generally speaking, in OChem, we don't do a whole lot of tricky math in this class, really, right? We're not really doing a whole lot of algebra or anything like that. It's mainly we're thinking qualitatively, what form is this going to be in? All right, the other way I can test this is rank them in terms of increasing acidity. So from from least acidic to most acidic. I don't like the wording, like saying it like this. I'll always try to write my problems so like from most acidic to least acidic. You just have to pay attention to that if you're taking a standardized test. That usually they would put the caveat with one being the most acidic or one being the least acidic.
Because in my mind, it's like, okay, in order of increasing, so one should be the least acidic.
Correct. Yeah. So I'm just gonna say most middle least.
Perfect. I like that so much more.
So butyric acid, also known as butanoic acid. So we're looking at all of them are the same parent molecule. We have 2,4-dichloro, 2,3-dichloro, or 3,4-dichloro. So we know that the effects of electron withdrawing groups, especially if we're just looking at what's the electronegativity, just straight up pulling through the bonds, they call that electron withdrawing through induction. If you're doing it through the bonds, it's called induction. It drops off pretty quickly the further away you get. Thinking back to NMR, right, in terms of shielding. So 2 chlorines as close as possible, we would expect to be most acidic. One on carbon 2 and one on carbon 4 should be a little bit less acidic. Probably pretty close though, right? Because they both have one chlorine on carbon 2. Small difference. This one's gonna be pretty significantly different than the other two.
And it's also methyl, I don't know why they did that.
Oh, because methyl's electron donating, not electron withdrawing, so it's actually, it's gonna be less acidic than the unsubstituted.
Yeah.
Because it's gonna look like. Actually, you can't have.
Yeah, what that's, yeah.
That's a typo, right?
So that's just 3-methyl
Pentanoic.
pentanoic acid, yeah.
I mean, we can obviously see why they did that, they were trying to make the point, they were
Yeah.
trying to make it all the same molecule, but.
Yeah.
So either way, because now it's not electron withdrawing at all, it's electron donating, we'd expect this to be less acidic even than just straight butanoic acid. By a little bit, again, methyl groups aren't that electron donating, and it's talking about a carbon 3. It's gonna be pretty close to the same pKa as butanoic acid. And you probably guessed by the root there, David, but we talked about the propionic is the old school way of saying propanoic.
Cool.
So 3 carbons.
Yep.
So 3-bromo. 2,2-dibromo or 3,3-dibromo. Most acidic,
Most.
least, middle.
Give me a second before I realized there were 2 separate lists. I was looking at them like, ooh, this would be interesting.
That would be, and that's, I'll try to only to not make too many variables changing at once with these type of questions, because it does get to be too much. Yeah, the main thing is that you keep all of your unsubstituted carboxylic acids are going to be in the high 4s unless there's resonance, and then they're in the low 4s.
Oh, okay.
Right, so resonance gets you down a little bit more acidic, but not by much, and if you use that as your baseline. You know, butanoic acid is gonna be awfully close to propanoic acid, which is pretty dang close to acetic acid. All right, as far as, or where we've already seen carboxylic acid show up, we saw them show up a lot when it comes to oxidizing things. So ozonolysis, remember the full name of that was oxidative cleavage. If you do ozonolysis of an alkyne, you get the carboxylic acid. Or 2 carboxylic acids, as long as R is more than 1 carbon. If you have a primary alcohol and you oxidize it with chromic acid, or trying to think what's the non-chromium based that goes all the way to the carboxylic acid. PCC was PCC. No, that was still chromium, huh?
Yes, it was chromium.
One of the
but
C's
it was
is chromium, but it was safer chromium. We have to go back and review what the exact other ones are, but again, standard is just chromic acid
I feel
or
like
sodium chromate, dichromate.
I always like memorize those, before the exam, and like I always forget them immediately.
It's as long as you know that they exist, you don't need to remember the exact title, right? As long as you know that you can do this reaction, you can always go look it up.
Exactly, yeah.
And you can also see how back before the internet was a thing, OChem was even harder because you were expected to still remember that because you had to memorize things.
But there are books for that.
Right. There was, to some extent, they were okay with that, but I had really old OChem instructors and they didn't say you need to have this memorized, they just expected it.
They just.
Like, as you all know.
Yeah.
That Joseph Mikkel was Eastern European guy. He's the one who threatened to fail us. We mispronounced Huckel's name.
Amazing.
He was also the one he would ask us some bit of review from physical chemistry 4 years ago. And then and when nobody had an answer for me, you have known this since kindergarten, yes? That was the attitude, the old school chemist is like, what do you mean? You studied this years ago. Why don't you have it memorized yet?
Right.
Oxidation of, we also had if we had dichromate on a benzylic carbon, as long as there was a hydrogen, we could get the benzoic acid from it, right?
And it has to be Sp3, right? Sp2 won't work on it.
It can't be,
It can't be an alkene, yeah, it has to be Sp3, it has to have a hydrogen. So with caveats, but we have seen these show up before. The other way that these show up that I think we touched on at the end of last chapter with the nitriles. So the nice thing about nitriles is that cyanide is a pretty good nucleophile, other than the whole exceedingly toxic thing. It doesn't cause any environmental concerns though, really, because it's a molecule most things know how to deal with to some extent. And it dissolves readily in a lot of different solvents. So it's a pretty easy nucleophile to work with. Just don't breathe it.
It's also a great song by Metallica.
There you go. First time I remember hearing about cyanide was from an old Agatha Christie book. I went through my parents' library when I was in 4th grade, read all the murder mysteries I could find. And Agatha Christie loves cyanide. Cyanide's in like half of the books she's written.
Amazing.
So if we just use cyanide as our nucleophile, we get a nitrile. And the nice thing about nitriles is we can convert them pretty readily to a carboxylic acid just by exposing it to water and acid. A little bit of heat. And that's a reaction we'll talk about in more detail, but it's a pretty easy way, especially if you want to add a carbon. Switch to we could do something like turn this or use hydroxide as a nucleophile and then do an oxidation. That would work too, right? But if we want the extra carbon in there, sometimes it can be convenient to use cyanide. If we want it not on the benzylic carbon, one carbon further away, it's an easy way to add one carbon, especially if you're gonna then if you want it to be a carboxylic acid as well. Then the other thing that happens, and this is what we were going to try to do with our Grignard reagents, but we couldn't find dry ice. Remember that?
Oh.
We were going to take our Grignard reagent. We had phenyl magnesium bromide. We were just going to expose it to dry ice as CO2, which is CO2, to add a CO2 group to it. Basically, you turn your Grignard reagent into your carbon into a strong enough nucleophile, it can attack the carbon in CO2. Which is not a polar molecule, but it has polar bonds, so there is a partial positive on that carbon still. And so that's another way that you can add, if you can make a Grignard reagent, which we would be able to do in this case too. We could do both of these, either of these approaches to make this molecule. If we started with phenylbromomethane, exposed it to cyanide, and then acid and heat, we can get this. Or if we make a Grignard reagent out of this and expose it to CO2, that would give us the same net result.
But how careful do you have to be to let them avoid there being any like water when you expose it to CO2? Because I know carbonic acid is a thing, and CO2 and water react, and then you get Grignard.
Right, so we don't. Traditionally, we've not had great yields for this reaction, and I think part of the problem is that if you have solid CO2, you're gonna get some condensation even in a dry environment like we live up here. So really, if you get decent yields, especially if you have a CO2 tank and you bubble CO2 instead of using the dry ice approach.
Yeah.
Because dry ice just pulls too much moisture out of the air. And when you have water, the Grignard reagent, that doesn't work very well. You wind up just adding OH here and turning your Grignard reagent into magnesium oxide. And that reaction happens really fast with high yields. To the point where if you just take a straight Grignard reagent and expose it to water, you might wind up with it igniting. So if you'll remember our Grignard reagent, that was one of the ones where we added it slowly with an ice bath to keep it from getting too hot.
Yeah, makes sense.
A lot of pent up energy in that.
Yeah.
GX1.
Let's do this before we take our break. Because then we're gonna start talking about other carboxylic acid groups. Again, this is more review. You have the acid, if you expose it to a strong enough reducing agent, you can turn it into a primary alcohol. So we can take that back and forth pretty easily. We played around with that when we first brought in alcohols, right? Oxidize the alcohol carefully, you can get it to stop the aldehyde, oxidize it like crazy, you get the carboxylic acid. Once you're the carboxylic acid, you can reduce it all the way back to the alcohol. So we had a pretty robust discussion. I think that was when right before we did synthesis problems back at the end of last term, right? You can also use boraine, BH3. In THF, you have to use a non-protic solvent, or else the boraine will react with the solvent. The boraine in THF, we can reduce acids without reducing ketones or aldehydes. So it'll reduce class 1 carbonyls, but not class 2 carbonyls. Boraine, much like sodium borohydride, it's a reducing agent, but it's kind of a gentle reducing agent. So it tends to be a little bit pickier about what it reduces and how. All right, let's take a break. Let's come back at 11 o'clock. And we'll talk about the other acid derivatives, more nomenclature, more functional groups.
What is the plan for lab today?
So I'm thinking today, how did your computational, have you worked on the computational one at all yet?
Not yet.
So it makes sense, we took some time to work on that over in the library, I'll just come over there and hang out.
Okay. That would be awesome. Yeah.
Yeah.
I just like need to be at 2x4. That's what I was asking. So.
Easy enough today.
Awesome.
Yeah, and we were talking a little bit too, check the recording for all the details we talked about, but labs for the rest of the class are gonna be mostly. So this is week 7, right? So if we finish our computational lab this week, that means next week's week 8 of 12. And really only out of 11 when it comes to labs.
Right.
So in week 8, we'll do a paper lab 2, we're talking about 2D NMR.
Okay.
Which is kind of a fun topic. It's a little bit different at least. It's not just assemble glassware, reflux, recrystallize and so we'll do that one next week, and then we'll spend a week. I think we're gonna try and extract caffeine from tea leaves. That one doesn't usually get great results, so it's kind of a fun one for me to give you a procedure, but hey, this probably won't work the first time, let's see if we can make it work. And then the last 2 weeks will be, so we'll spend one lab period picking a research paper for you to read, and then the week after that, you'll present it.
Oh, cool.
So I'll help you pick something that's first of those weeks will be like, here's how you find paper, because that is that kind of skill, like the difference between a review article and etc. So we'll talk about picking your article the first week and then we'll present it the week after that, and then it'll be finals week.
Well,
Boom.
Yeah, so we don't have a whole lot, no more wet chemistry labs, I don't think. Wow. I have one we can do, but I think the other stuff is gonna be more useful,
Yeah, sweet.
okay?
And then we will have our completed, probably not really completed, but semi-completed OChem.
Wow, that's crazy.
It's years gone fast. All right, since that was, I'll still count that as talking business, so let's come back and talk.
Thank you.
It's like when, when the boss writes off a meal as a business expense, right? We talk business there.
The gym, I just been like.
What you been up to lately?
Well, I've just been down at Davis a lot,
Oh, okay,
since my sister goes there. She
gotcha.
was like, you know what? I'll just take you like to my classes and you can just become a student for like a few days.
Sweet.
Fuck it.
Yeah, that's cool. It's a preview it basically.
Yeah, that's pretty much first from us or else. So,
I mean,
sure, yeah, it's just,
it's more of this.
it's just.
Yeah, the biggest thing I found, and I don't think you're gonna have a problem with this, is just go to class.
There's just fucking go to class.
Yeah.
Because,
And just be there and be present.
yeah, no, that's what my sister told me, she was like, you know what, like, you might wanna have years, just like not show up, but like, don't fucking do that, like, that's the worst.
Yeah, then you just fall behind and now you're just done. Yeah. No, that's that was the problem I ran into in college. I was so burned out, couldn't know what I was gonna do. The course load was like insane. I was just like so tired of it. So I ended up volunteering with the fire department and the ambulance for my
There
mental.
you go, that's how you started that career out, huh?
It is, exactly, and but I spent way too much time doing that, I was probably putting in at least 40 hours a week doing that. And so was maybe not going to classes as much as I should. Got a degree, so I'm like,
There you go.
that worked out, praise the Lord, but other than that. Yeah, no, definitely, like I think I got like a C in OChem in college. That's why I'm taking this, I'm like, if I'm gonna go, I don't know, like, I'm thinking about medical school. And like praying about it, and I'm like,
No, dude. Are you fucking kidding me? You're smart.
oh no, it's not a matter of being, like, I know I am, thank you, I appreciate that. Like, I know I could do the coursework, I'm not saying that in like an arrogant way, I'm like, I know I can do it, it's just.
No.
But like, at the same time, like, so many of the doctors I know are just like. Like, there's a small handful I know that are happy, but I'm like, yeah,
The inchification of
yeah.
the healthcare system is a real thing.
Well, that's what I'm like, Barbara, I'm like considering also like a PA and NP.
Yeah,
Especially
Yeah.
like I have my bachelor's, so it'll be like a year and a half for me to get my PA, and then I can have my own practice 15 minutes down the road.
right.
I think that would maybe scare your sanity at least a little bit.
And yeah,
Yeah.
I'm debating and praying about what I need to do
Yeah,
going forward. Like, part of me still wants to do medical school, and part of me likes having a life.
well, the other thing you can do is you could go, if you like if you don't want to deal with the medical system, but you like studying the stuff, you could always go to grad school. Do PhD doctor instead of medical doctor.
No, I like.
You
I
don't
like
get
medicine.
paid as much, but
Yeah, I like the medicine.
Yeah.
I enjoy studying stuff too, but like my passion
is the people,
patient care.
yeah.
So. But yeah, no, I thought about that too, because I know there's some programs where the MD PhD is like pretty much is, if you get in, it's like you don't have to pay nearly as much.
Yes.
There's like good scholarships and stuff. But I'm like, that's also just an insane amount of school.
Well, and that's not setting you up to be a practicing
No.
physician. That's setting you up to be a research physician.
Exactly, especially because you do all of your clinical
Mhm.
education first, and then you're like, cool, finish that, let's spend years doing a PhD, and then it's like, then you go do rotations after you've already forgotten everything.
Right, which is a good way
Yes.
to bring, I had a friend in grad school who was doing that route,
Yeah.
and it worked. It worked pretty well, but it was definitely geared towards, you're not going to be a practicing physician, you're going to be setting up, designing clinical trials and doing research more so than at taking care of patients. Which, if that's what you're into,
Been great.
it's a really cool program, and there's not a whole lot of more prestigious, like,
No.
well-regarded programs than MD, PhD programs, but it's a lot.
That.
right, and then you can do it all.
Slavic / Asian parents will be so proud. Doctor Esquire, yes. I don't know, there's not like a dedicated like sports clinic. Like Barton was really good at doing ortho, but there's not like a dedicated.
There's PT, there's a lot of PT
For sure.
clinics.
But I mean, like a place where it's like not just PT, but like there's doctors, surgeons there. they do like.
Oh, right.
Like joint stuff that are like, and that's just like what they do.
It's in Zephyr Cove.
Is it?
Yeah, there's
Okay.
well, there's one anyway.
Okay.
It's some of the same doctors that work at Barton, but they do. When my daughter broke her elbow, that's where they took her like to do like the specialist x-rays, because on a 8-year-old with a broken elbow, it's really hard to see any sort of break on a regular x-ray. So they took her in there because they had the specialized x-ray equipment and specialists interpreting it.
Yeah, green stick.
It was a green stick.
Yeah.
It just bends, it doesn't.
Yeah. So, but there's, you know, who you could talk to. I don't think you've had him for a class, but Carl, or the other, his wife is an OB at Barton. So if you're interested in hearing what the local landscape is like, Barton's kind of got their fingers into all the medical stuff in South Tahoe. So if you want to get an idea of what that's like, he's a good person to talk to about that. All right. Thanks for bearing with me. My oldest is out sick again. I think maybe he has a migraine this time, but his baseball team is depending on him for tonight. And so I'm trying to like, well, maybe he can play. He went home sick from school, but if he sleeps for 5 hours, then maybe he'll be ready to play baseball.
5 hours, popping separate.
Yeah, piece of cake. Stick with it, kid. All right, so these other functional groups that are considered acid derivatives, they all have the same general form, like we talked about at the beginning of aldehydes and ketones, where they're you've got a carbonyl with a good leaving group attached to it. Good leaving groups are more electronegative than carbon, so they all have the same oxidation state. And these four in particular, these 4 plus acids are all really easy to convert back and forth between. It's a very similar mechanism to go from any of these. But they do have sort of like a ranking of reactivity. So if you want them to be more reactive, you take your acid and you turn it into an acid halide or an acid anhydride. If you want to be less reactive, you turn it into an amide. So you might notice as well, since we've been talking about biochemistry a lot here, amides also include if you put another carbon on that nitrogen, that's still considered an amide. So an amide is an amine attached to a carbonyl group, which biochem has its own name for that type of bond, right? Peptide bond. So peptide bond is an amide bond. In chemistry, we call it an amide linkage. In biology, they call it a peptide bond. Same thing. Potato, potato.
This is like primary, amide, secondary.
Yeah, so we would so there might be something funky about the way they use the terms primary and secondary when it's amide. Because we would definitely, so
I was thinking amines, right?
right, so that's a secondary amine.
Yeah.
I'm not sure if we use the same terminology for amides or not, so hold off on that.
Yeah.
And then the last one that's kind of like the persona non grata, still technically an acid anhydride because the oxidation state. Nitriles are really kind of their own thing though. It doesn't have a good leaving group, even though it has the same oxidation state as these others. And you can like we just saw, you can convert a nitrile into an acid. And then that puts you into this ballpark, and all the reactions back and forth between here are all fair game. But so that sometimes you see nitriles included as acid derivatives, but more frequently they're kind of their own thing. Especially since it doesn't have a carbonyl attached to it. All right, when we're naming these, we're gonna name all of them because they're all pretty much the same. Acid anhydride, sorry, acid halides. Name it like it's the acid, except that then you switch the ic acid. And you put YL, and then whatever the halide is. So acetic acid, as in acid bromide, becomes acetyl bromide. Benzoic becomes benzoyl chloride. If you're using the IUPAC names, they pretty much all have the OIC, but you only drop the IC part, so you get this weird OYL suffix at the end. But what we'll see is that they pretty much all of these, we're gonna be basing them around the acid name. So as long as you can name the acid, you just tweak the ending of the acid. So nitriles, you just drop ic acid and you put nitrile on there. Acetic acid becomes acetonitrile. All right, so some vocab to memorize. But we've been laying the groundwork for all these. These aren't unfamiliar groups to us at this point. Anhydrides are named by replacing the acid with anhydride. So acetic acid becomes acetic anhydride. You do get some funky ones here, like the dioic acids, if they're long enough, you can make an anhydride that's a ring. So this one winds up being used a lot because it's relatively stable and doesn't have any extra atoms. So a succinic anhydride is this weird 5-sided ring because succinic acid has 4 carbons. And basically it's these anhydrides, it's basically like you take 2 of the acids and do a dehydration reaction. You lose an OH from one side and you lose an H from the other side. So you take OH away and you take that H away and connect the oxygen over to the other carbonyl. If it's an asymmetric anhydride, these tend to be pretty rare. Basically because anhydrides are exceptionally reactive. So most of the anhydrides that you can buy that have a decent half or shelf life tends to be symmetric anhydrides. Just because if they break apart, they can reform back into the anhydride form. Acetic benzoic anhydride is going to be, a little bit more tricky to keep on the shelf. All right, so those are the easy ones. Amides are a little bit trickier because we can have something attached to that nitrogen. So this is one of the first times we've seen substituents that we would name as a branch, but attached to something that's not a carbon. So any substituents that are attached to the nitrogen, we just specify, instead of putting a number to it, we use N. So N dimethyl acetamide. But you could also have, 1N dichloroacetamide. So you can mix the N in, just use it like it's a number, basically. If you ever have a molecule that has 2 nitrogens, you basically are going to say N1 and N2. But yeah, same way we name these, name the acid, drop ic acid, and say amide. And then last but not least are esters. And the esters are the trickiest because the esters always have 2 sides. And they're not always symmetrical. In fact, esters are never symmetrical, kind of by their definition, because you have this carbonyl on one side and then an oxygen and something else attached. And so the way we name these is this, the side of the molecule that has the carbonyl is the dominant, is the parent molecule. And so, and then you just name whatever's on the other side of the ester as a branch. If you're really careful looking at these, usually OChem textbooks like to put branches all as one word with the parent molecule, right? Which is why it looks so confusing. With esters, they put a space in between. So ethyl acetate, the fact that there's a space there is telling you that the ethyl group's on the other side of the carbonyl or the other side of the oxygen. Not that there's an ethyl attached here. So subtle differences, but chemists love those, right? We can communicate so much without saying a thing. We're saying as few words as possible, but it makes it really confusing. And so the key though with the esters is you find the parent molecule, name your parent molecule. The parent molecule is the side of the carbonyl. Even if the other side is longer. So it's not about your longest continuous carbon chain, it's about your longest continuous carbon chain that has the carbonyl. So since that was a whole bunch of functional groups all in a row, let's do some recall. And it can be helpful to start by just figuring out what the functional groups are, because that might trigger something in your brain to remember, oh wait, with acid anhydrides, I just say anhydride. So 3 carbons, symmetric. So propan, let's see. Propanoic anhydride. So we got an amide, so it's gonna be propanamide. Diphenyl. And they're on the nitrogen, so N diphenyl propanamide. Let's do F before we do C. Just so we can get practice with a simple ester before we do a diester. So this is the side with the carbonyl, so that's our parent molecule. So butanoate. And we got a propyl on the other side, so propyl butanoate. And keeping it as 2 separate words, again, seems like a really nitpicky thing, but that allows us to use all of our regular rules when it comes to branches. If we don't have simple straight chains here, we need to put a methyl. That's how we're going to differentiate between is the methyl on the other side of the ester or on the parent molecule side of the ester. Then there's a common name for this dioic acid, but it doesn't matter. I don't remember it, so I'm not going to worry about it. I'm just going to name it as 1, 2, 3, 4. So it's butane dioate. So because it would be butanoic acid if it was just the acid form, right? But when it's an ester, you drop the ic acid and you name it like it's the deprotonated form. So it's butanoate, or sorry, it's butane dioate. Because it would be butane dioic. acid, if it was the acid. So we drop the acid, dioate. Looks like there's entirely too many vowels for that to be a real word, but it is. And then we name it's both sides are the same, so it's dimethyl. And we're starting to get into the realm here where, like, if you're in the real world and you messed up your rules for this, like, nobody's gonna look twice at you because, like, we're getting into some pretty complex gray area. Yeah, like, well, is that dimethyl on? How do we apply it? And so it would get even trickier if there was a methyl there and a methyl there, because that would be dimethyl. Right, so it's one of those where you do your best and try to make it unambiguous, and it doesn't matter if you get it 100% right, as long as the person reading it can get to the right molecule.
Carboxylic,
Not a carbaldehyde because it's an amide.
right. Carboxylic.
So I would actually name this as methanamide.
Yeah.
So it's gonna be 1-cyclobutyl. 1-cyclobutyl, N-methyl. N-ethyl. N-ethyl, N-methyl, 1-cyclobutyl methanamide. Again, there might be another way, like carbaldehyde, there might be another way to name this rather than going methanamide. But if you do it this way, somebody reading this would be able to get to the right structure, even if there's a different way that also works. And why the IUPAC names are the way they are and why we focus on those so much is because they tend to be the most powerful, most universal.
So the methanamide is referring to the carboxylic acid and the nitrogen. Okay,
Yeah.
just wanna make sure.
Yeah, we're saying it's the parent chain has more carbon in it.
Mhm,
So the cycle group.
right.
Okay, it's not so like-
Okay.
How about a simple one? The trickiest thing with the nitriles is, do you count that?
I want to say for like propane and nitrogen.
Or is this propane nitrile or is this butane nitrile?
Well, I feel because the CN is the nitrile group, I feel like it wouldn't count. This is my like intuition, but that could be completely wrong.
So that's acetonitrile.
So acetic acid turns to acetic, so you do count the carbon on the nitrile as part of the carbon chain. So if it was an acid, it'd be butanoic acid, so it's butane nitrile, butane. Is there an O on there? O nitrile. Yeah, O nitrile. Butanonitrile. So this one, I would use the common name for it just because it's a unique sort of molecule. That's the succinoic anhydride. So succinate is 4 carbons, is butane dioic acid. The common name for it is succinic acid. So you could name that, because one of the reasons I like that using the common name for this is because otherwise you have to deal with, well, how do I say that it's part of the same molecule? So succinic anhydride. But again, I'm not gonna test you on ones like this. This is just a common enough molecule. We actually use it in lab fairly often because it does have a decent shelf life despite being an anhydride. But if we're counting 1, 2, 3, there's succinic acid.
Mhm.
You take that and you fold it around on itself, you get succinic anhydride. Maleic anhydride does show up fairly often as well. If I go back to the common names. Malonic acid, maleic acid is not on here, so never mind. I must be thinking of something else. Oxalic acid, you can't really make an anhydride with it, it's not enough. Actually, did I see? 1, 2, 3, 4, 5, yeah, so 1, 2, 3, you have to make a 4-sided ring to make the anhydride out of this. 3-sided ring here, so obviously we're not gonna do that. So oxalic anhydride would be something different. It wouldn't form that ring structure.
Imagine it'd be 2 of them would come together and go through a dehydration.
Except that then you still have an unreacted carbonyl group on their carboxylic acid on the other side. So I don't think oxalic anhydride is a thing.
Is there any way you can like memorize that? Anyway?
You could do like meth or acetyl oxalic anhydride. Acetic oxalic anhydride would be so there's your oxalic acid in the middle. There's your acetic acid and acetic acid. So, like I said, that succinic anhydride shows up relatively often because it's a simple molecule that's also an anhydride without having this aspect to it.
Yeah.
Went too fast. So more practice with the esters, because the esters, like I said, they show up all over the place and they are a little bit finicky. Most of these other ones are fairly specific molecules. Esters show up everywhere. Esters show up all the time in food chemistry too, because they have very distinct flavors. If you've ever had a Belgian style beer that had like a fruity flavor to it, or even like a wheat beer that had like a banana odor to it. That's the ester that the yeast produces as a byproduct. Certain strains of yeast produce certain esters as a byproduct of the fermentation process. They can give you some weird, fruity, funky flavors that have nothing to do with fruit. A Bavarian style Hefeweizen in particular uses a strain of yeast that if you ferment at, I want to say if you ferment at 68 Fahrenheit, you get eugenol, you get clove flavor produced very subtly. And if you ferment it 3 Fahrenheit higher, 71 Fahrenheit, you get banana flavors instead of the clove. And if you ferment right in the middle, you get both of them, but in a really weird kind of subtle way. It sounds like banana and cloves, that seems like it would be off-putting, but it's actually pretty good. They go pretty well. So esters wind up being important in a lot of areas because they're easy to make, they're simple molecules, and they have very distinct flavors. So most artificial flavored candy as well. Like the banana flavor from Banana Runts. Like everybody knows, like, or the noun laters, the banana noun laters had that really specific flavor to them. That's an ester. It's the same ester that the yeast produces in the beer. Just in different amounts.
No, this one's weird because it's like, I want to say it's like a methanoate. Oh, but it's
Except the
phenyl.
parent side has the carbonyl.
Right, so but it's like there's just so this is just a phenyl, the carbon of the
So
carbonyl.
if this was just the acid, we'd name it as benzoic acid.
Yeah. Oh, okay,
So we're gonna name it
Gotcha.
as methyl benzoate.
there we go. Okay, that makes sense.
The trick, like the reason that we do the acids first, all of these other functional groups, their names come back to the acids. When in doubt, name the acid. Cool. And adjust the name from there. So because I is the same atoms, just with the carbonyl on the other side of the oxygen, right? So this one. Now that's our parent molecule.
Gotcha,
So that's acetate.
gotcha, yeah.
So that's gonna be phenyl acetate. And over here, and again, just to reiterate. I'm not just being careless by putting a space there instead of hyphens. Usually I use hyphens, right? Making it a space in two separate words means we can add prefixes on to either of these as necessary. So like if I did that, that's going to be methyl benzoate. But where the space is important, right? Because actually that's a good one. Let me white out the screen. We'll draw them all. We'll practice doing that. So that's not benzoyl chloride because there's an extra carbon in that, right? So our parent molecule is going to be the methyl benzoate. Parent molecule is right here. And, or sorry, I didn't put the methyl on there. Did it there. So that's going to be 2, 3, because that's carbon 1. And then the other part.
Do we need an oxygen, another oxygen?
Yes, sorry, that's where it is. And then we have chloromethyl as a separate word. If you're doing this in your handwriting and you're not sure if your handwriting you can differentiate between is there a space there or not, you can always use an underscore. But you just need to do something to make it clear that that's two separate words so that you know that the chloro applies to the methyl on the other side of the ester and the 3-methyl applies to the benzoate.
Yeah.
I know I like to go back, think about back in September, October, when we were first learning nomenclature, but.
Oh, she's beautiful.
Well, if I told you then, like, oh, in 6 months, we're still gonna be talking about nomenclature, you would have torn your hair out. But we build up to it nice and slowly, right?
Yes, progression is good.
In pedagogy, we call that scaffolding.
Nice.
Before I just give you that molecule the name, we have to build up to it. All right, going the other way. Dimethyl oxalate. So what type of functional group is that going to be? Ester. So oxalic acid was 2 carbons, both carbons are carboxylic acid, so. So you see oxalates as esters fairly often, not as anhydrides. Because that's a reasonable-ish molecule. On this, so they're using carboxylate, cyclopentane carboxylate, so it is similar to the carbaldehyde. Like we talked about, so we could have named this one that way,
Okay.
named it as carbamide, carboxamide. So phenyl cyclopentane carboxylate. Cyclopentane carboxylate. It's gonna look like that. And then it's phenyl on the other side of the ester. Again, though, if you said cyclopentane methanoate, phenyl cyclopentane methanoate, you could still get to that same, you named it as the methanoate instead of saying carboxylate.
Yeah.
Gets you the same approach, right? And methylpropionamide or propanamide, I mean. I always have a tendency to lose the syllable in there, so again, helpful to say the name of the acid,
Too many bells.
right? So 1, 2, 3, there's our oxygen, there's our nitrogen. And methyl.
Yeah.
And propanol chloride or propionyl chloride. 1, 2, 3. Carbonyl Cl. Yeah, the good news is that this is, I think, is the last functional groups that we're adding. So, which means for the most part, that's the last nomenclature. There might be a few things here and there as we start getting into some of the bio side, but this is the last IUPAC organic nomenclature that we're adding for the class.
Thank God.
All right. So last concept, we're gonna do this and I know that we only have 10 minutes left. But I want you to see this once so that when we review it on Thursday, it'll be reviewed, not seeing it for the first time. Acid derivatives. The reactivity of acid derivatives is based on how good of a leaving group you have. The best leaving group makes the most reactive acid derivative. So our acid chlorides, acid bromides are going to be better leaving groups, therefore more reactive than acid anhydrides. Because if you think about an acid anhydride, this whole thing is the leaving group then. And that's a stable molecule on its own. Much more so than an ester. Because if you think about a carboxylic acid or an ester, now our leaving group is just a deprotonated alcohol. Or if it's the acid, our leaving group is a hydroxide. We know those are fairly strong nucleophiles, fairly strong bases, so they're not going to be very good leaving groups. So typically, the most reactive is going to be the acyl chlorides, then acid anhydrides. Esters and carboxylic acids are roughly the same level of reactivity, but we usually rank esters as being more reactive than carboxylic acids because there tends to be more hydroxide or water around. So just based on concentrations, we wind up making the acids more often than the esters. Unless we are doing this in a solution with an alcohol. If we're doing this in water though, we're always going to have more of the carboxylic acid than the ester. So we think of the esters as being more reactive. And then least reactive is our amide. That NH2 is a really bad leaving group. Deprotonated amides are really, really strong bases. So once you make an amide, it's always easier to go downhill in energy than uphill in energy. So what we're going to see is from an acid chloride, it's really easy to make any of the others. From an acid anhydride, we can make any of the others that are downstream. But we have to get creative when it comes to making the acid chloride. So there's basically it's a lot of the reactions and the syntheses we're gonna see with the acid or with the acid derivatives are make the unstable form and then let it react to the stable form that you want. All right, and they're pretty much always gonna do the same thing. It's the same mechanism every time. This is nucleophilic acyl substitution. So now we had nucleophilic substitution, SN1, SN2, we had aromatic nucleophilic substitution, now we have nucleophilic acyl substitution. And this is different than the nucleophilic addition from class 2 carbonyls. Because we reform the carbonyl. It's the same first step as last chapter, right? Nucleophile attacks carbonyl carbon. And you make a tetrahedral intermediate. Then once you make the tetrahedral intermediate, what's different about the class 1 carbonyls is that leaving group. If you have a good leaving group attached, your leaving group leaves and you reform the carbonyl. Right, so this should look like a really familiar, kind of fairly simple mechanism. The only new wrinkle is leaving group leaves and we reform the carbonyl. And so that's going to be the way that we can go back and forth between these pretty easily. Because if you start with an acyl chloride and you expose it to hydroxide, hydroxide is a good nucleophile, right? And it's going to come in here and attach. And then the next step is your chloride's a good leaving group. So your chloride leaves, you reform the carbonyl, and you just converted your acyl chloride to the carboxylic acid. All you have to do is switch out what the leaving group is and what the nucleophile is to get any of these converted back and forth, right? Pick your nucleophile carefully, pick your leaving group carefully. And it's all the same reaction. Just like though with generating the electrophiles, to get upstream, we have to get creative sometimes. We have to generate a specific type of nucleophile that's strong enough that we can force something like a carboxylic acid to go backwards to get to the acyl chloride in the first place. But that's just like we did with the electrophilic substitution, generate the electrophile, generate the right nucleophile, and you can get that to work.
So then from like a carboxylic acid amide, you can use like NaNH2 and NH3.
Yeah, or just NH3.
Oh, that would work, yeah.
Ammonia works on its own
It's basic.
because it's basic. It's already got one pair.
Yeah,
So yeah, getting to amide is really easy. Expose it to a little bit of nitrogen.
sweet.
But that's also why peptide bonds are used is the wrong word. Are so common in biochemical systems. Is they're more stable than the rest of these. But on the other hand, it's actually pretty easy to take it and go backwards if you do it the right way. So enzymes can break peptide bonds, but it requires effort. So when you make a peptide bond, make a polypeptide, it's going to stay as a polypeptide until the cell or something takes effort to break it apart. So we think of them as being permanent. They're not permanent, they're just permanent enough. All right, let's end there. And then you guys want to work over in the library on the computational stuff or in the lab here? Either way works for me.
Yeah.
Library is a comfortable place to work, right?
Yeah.
Okay, so I'll meet you over in the library around 1.
Great.
And hopefully I'll have tests graded by then and hand those back.
If not. Oh, short. Islands.
Cool stuff.
Yeah.
That was, so we were talking about that